Welcome back everyone. In this session we will continue with step 1 of our workflow, which is data retrieval, but this time we will focus on NCBI GEO. GEO stands for Gene Expression Omnibus. It is the largest public repository for functional genomics data. So here you will find the data set for microarray RNA sequencing study and many other high throughput experiments. Let's see how to find and download data from NCBI GEO. First, search for NCBI GEO and click on this first link, that is Gene Expression Omnibus. This is how homepage of GEO looks like. Here you can find dataset by two ways. Either you can search by using keywords or GEO accession ID, that is a particular identifier for GEO datasets. So to search with the keywords, simply type the keyword of your interest. Like at this time I want to search for a gastric cancer data set. So if I type gastric cancer, so here it gives you two options. So we want to search the data sets. I will go with this first one, that is search data set from GEO data sets. Because we want to obtain the data set. GEO now shows me a list of studies. So each entry has a title, a short description, the title platform used, and the number of samples. Expression profiling by high throughput sequencing, that means it is the sequencing data. And for microarray data, we will have expression profiling by array. Here on the left hand side you can find out the different filters. Like you can select study type. We are interested in microarray, so we can select expression profiling by array. You can select other study types as well. You can also select the origin of your dataset, like you want a tissue samples or a strains or blood samples, whatever you want. You can specify the attributes here. Also, you can select the organism of your interest from the right hand side. If I click on one of these entries, I go to the dataset study page. On this page, you can find the information. First is this GEO accession ID. This is the unique identifier for a GEO dataset. Next, date of publication, when this data made public first time. Then the study title, organism, and experimental type. Expression profiling by array means that it's a microarray data. Next, we have the brief summary of the study and the overall design of the experiment. GPL is specified for the platform of GEO database. So the most common platform for human microarray dataset is GPL 570. Next, we have the detail for the each sample, and the sample IDs starts from GSM. If I open any of this sample, here I will find the information regarding each sample. And at the bottom of this page, we have the section for these supplementary files. Here you can find the links to download your dataset. It is a microarray dataset. So to download this dataset, you can click on this HTTP link. So when you click on it, the download will start automatically. And these are the .cl files. You cannot open these in your Excel sheet. So to open and to read these files, we will use R for further analysis. So at this step, you can only download the dataset. For further processing, we will use R. Or if you want to download the specific sample, you can select this custom option, and from the list you can select the samples of your interest. Again, we need to download the metadata sample information. So you will get these information from the series matrix file. On NCBI GEO, you can get the sample information from the series matrix file. We will download it directly from R to access metadata and feature data, which we will use in further analysis, and I will explain there. We can also download the raw files directly from R, but the problem is that when we are start fetching the data, for that these files are too large. So if your internet connection is distorted, the download will fail. So it's better to download raw files manually, because these are the large files. So as I said before, you can search dataset by using keywords or using accession ID. Like if I search for the particular accession ID GSC 7793, so it will takes me straight to that data set. This is the quickest way to access a study if you already know the ID. But how you find these accession IDs for that particular data? Here is a useful tip for you. These accession IDs are very often mentioned in the research articles. So to find them, we can use Google Scholar. If I search by using keywords like breast cancer and gene expression, use these Boolean terms results to find out the exact results. And I'm interested in microarray data, so gene expression, microarray. The next important keyword is NCBI GEO. So this will help you to fetch out the data set. So this gives us a list of published studies. If I open one of them, like if I open this first one, usually you can get a session ID from the abstract section, methodology, or data availability statement. In this study, they use one microarray dataset with the accession ID GSC 14776. So in this way, you can find out the accession IDs. Sometimes you find the multiple accession IDs. Similarly, you can find out the accession ID for the Array Express as well. So instead of NCBI GEO, search for Array Express. Again, click on this first link. So in this study, they use the microarray dataset from Array Express database. So this is the accession ID for Array Express data. In this way, you can get the accession ID for NCBI GEO or Array Express to find out the particular data. You can directly find out dataset from their databases, from these databases, as well as by using a literature search, you can also get the accession IDs. Again, keep in mind when choosing dataset, look for datasets with two biological states, such as disease versus control. You are open to work with any disease type you are interested in, is either it's a cancer, heart disease, or any other. It should be from two classes: normal versus disease versus healthy individual, not from the subtype of red disease. We will use the same dataset for machine learning purpose. So for machine learning, we will focus on a binary classification task. For that, we need only two classes. Second, try to select datasets with more than 30 samples. So that's all about the data retrieval. In the next session, we will proceed with data normalization step. Thank you.